Yeah, sure. Thanks everyone for having us. This presentation is piloting into future publicly available R-based submissions to the FDA, which dovetails nicely, I think, with the Roche and Novo Nordisk presentations that preceded us. And I'm from GSK. My name is Ben Straub, and I'm here with Joelle from Roche. And our next slide is just a little bit about us, different initiatives that we've been involved in case you want to read a little bit about us and what we do in the R landscape. But we'll skip that and just jump right into the meat of the presentation. So today we really wanted to talk about some of the challenges that we see for why we don't see wider adoption of R-based or multilingual submissions, and why we think to kind of remedy that, why we think publicly available R-based submissions are important. We'll go into a little bit about what we've done for these publicly available R-based submissions, like what kind of TLFs, atoms, that kind of stuff we've created. And then at the end, we'll kind of close with like what we think is on the horizon, working with containers, working with WebAssembly, dataset JSON, and hopefully make a pitch for folks to get involved. So I'm going to move on to the next slide. Yeah, so for me, like as a programmer working on studies, to me, I see some challenges with just how many packages that are available out in the R landscape, which is, I think, very exciting, but at the same time can be very overwhelming, especially for new users, especially for users who have really intense deadlines. And some of the remedies that have been put forward, which I think are very helpful, are things like snapshot environments. The Pharmaverse initiative, which I think has a ton of promise, is developing packages that are scoped to clinical reporting needs, and Open Statsware, similar initiative for dedicated stats packages. The question of like how do we validate some of these? Oh, I'm hearing some reverb. How do we validate some of these packages behind the scenes? I think CRAN is a great package repository, but we kind of need a little bit more validation. So things like Validation Hub, there's some great white papers out there that deal with that. Another big challenge, not only with the options, but just the software compatibility. A lot of programming stats departments at the different pharma companies use Linux, whereas like the FDA uses Windows on their machines. So that can be a bit of a challenge. Like, how do you get around that? How do you get around all the package versions that are coming out constantly? Which, it is great to see things being built and reimagined and iterated, but the versioning can be a pain point. And then I think another, and this is where we'll move into the R-based submissions, publicly available submissions, is there's not a lot of there's some great talks and information from Roche and Novo Nordisk who are pioneers in this space, but I think having just being able to see the code and see the minutes and see the ADRG can be very helpful. And I think this is where the submissions working group plays a big part. So next slide. So we have this terrible acronym, RCRSWG. That's our Consortium, our Submission Working Group. We work with the R Consortium and to advocate for the use of R. The Submission Working Group, which is part of our consortium, is really focused on helping to show open examples of submissions, so that later on to make it easier for those who come after us to have an easier time with doing submissions. So we do everything out in the public. We do everything so people can inspect and learn from, which I think is really important to pushing more our adoption for the submission space. Next slide. And it is an open working group, so anyone can come and join. Lots of pharma and CRO companies have joined up and make small contributions or help lead entire efforts. And then people from academia have also joined. So I strongly encourage folks to join. We meet once a month at in a public meeting for about 1 hour a month. So And then it's kind of like choose your own adventure, like how much do you want to get involved in? So next slide. And the submissions, and we're going to go over in detail like what each submission is consisted of, but we've really been focused on the FDA and the eCTD portal. But we are interested in working with other agencies. But FDA has just been the one that we've been focused on and had the most reception from. We kind of use the same data over and over again, just the CDIS pilot data, which has been nice to kind of maintain consistency between the different pilot submissions that we've done. And like I said, we keep everything out in the open. And how we do that is we have a lot of different repos dedicated to each pilot. Typically, there's a development repo where we do all our code development, and the submission repo, which is the repo that's dedicated to moving the information over into the FDA. And then we have various utilities repos, which we'll talk about one later in the pilot 3 information. And again, all the meeting minutes are available for you to review. You can see feedback we receive from the FDA, discussions that we have on pain points, all that good stuff is up on our website. Next slide. Yeah, so the submission working group, we've done 4 pilots. The first one, just some simple TLF table listings and figures R scripts, and it used something called a package light. That was submitted and reviewed by the FDA, completed, and was very exciting. We'll go over a couple little pieces of that in just a second. Pilot 2 was a submission of an R Shiny application that just had the same TLFs, but just as an interactive data display. Pilot 3 and Pilot 4, Joel will take over and kind of go over what we did that was a little bit different from Pilot 1 and Pilot 2. So next slide, Joel. Yeah, so the pilots are very simple. So Pilot 1 was just 3 tables, 1 figure, not a lot of, very basic. We used the CDIS pilot data that was made in SAS to kind of test things out. And we made use of these, of the REMV package to help with reproducibility and stability. And we made use of this package light package to help package everything up into basically one text file and submit it through the portal. We'll talk about that a little bit more in Pilot 3 and how we differed a little bit with that. But you can see here when we submitted the package, we got some feedback, and after a successful review, we considered that pilot completed. And the findings for that kind of, or next slide, Joel, findings for that package or for that submission were very minor. There was a couple issues with they just wanted some information about the ANCOVA model that we used. They noticed that some values were off due to rounding, and just a couple of the column headers seemed to be mismatched. So very minor details. Joel was nice enough to make a QR code. If you're interested, you can actually use that QR code to get a link to our GitHub repo, and you can see all of the information that's there for Pilot 1. Next slide, Joel. And so Pilot 2, again, very simple, just 3 tables and 1 figure, but now there's a twist where it was all bundled into a Shiny app. And I think this is an exciting way to kind of present data to a reviewer at a regulatory agency. Again, it's just basic data set filtering functionalities. The source data, again, was the CDIS pilot data. And the submission was done in November 2022. And there was a lot of discussion about some of the potential issues with the Shiny app. So there was a lot of back and forth, but eventually the FDA considered it a successful review, and we completed it all in September 2023. And then the findings slide. They just had a few potential kind of findings around the concern for p-hacking or cherry picking subgroups. They did kind of recommend that keeping things more static for certain tables is important. They really like the interactive features for the KM plot. They also wanted some more documentation around potential warning messages when the FDA recreated the Shiny package on their systems. And so we started documenting all that kind of stuff in the ADRG, which becomes very important for Pilot 3. And there was some discussion about trying to move away from using package light, which we had used in Pilot 1 and Pilot 2, and just seeing if we could just submit .r files through the portal. And there was some concern also about having all the packages on CRAN rather than trying to submit them through the portal. So that was pilot 1 and pilot 2. Again, the focus is really on the process, less on what the content, just seeing that we can move these type of R files to the FDA. So now Joel will take over and start us on the pilot 3 and pilot 4 journeys.
Well, yeah, thanks, Ben. Yeah, I was really excited to see the progress that was made from pilot 1 and pilot 2, and coming from that announcements and what was going on in the world of in our industry and trying to submit open source R, it really triggered me to want to get into working with our consortium. And so I helped co-lead the pilot 3 study, and just wanted to give you guys an overview of kind of what we did there, coming from pilot 1 and pilot 2. So pretty much in this pilot, we focused on the ESA package, which again leverages the SCTM data from CDES pilot 1, as you see here. This data is publicly available and simulated, and we're using it to help then generate the atoms for pilot 3. So yeah, this was pretty much our main focus in pilot 3. So atoms was the main focus, and then the TLFs using our atoms to then regenerate the outputs that were made in pilot 1. So again, the complete study package here includes the CDES data, SCTM data from CDES pilot 1, the 5 atoms, which you see here, ADSL, ADAE, ADLBC, ADADAS, and ADTTE. Again, the note here is that atoms were primarily generated in R using R scripts. The package that we use is pretty much available across Pharmaverse now is Admiral. So we use this in our pilot. Again, the 4 TLFs consisting of 3 tables, 1 figure. These are all also regenerated in R, derived from the pilot 1 scripts. And as Ben alluded to, so in this pilot, we installed and loaded the proprietary pilot 3 utils R package into a zip file. And this package is essentially what we use to help the FDA review, ensure reproducibility and accuracy of our analysis. And, we didn't submit it using package light, as again, we wanted to try all alternative approaches to submitting a package, and we were able to do it in a .zip through the ECTD portal. So yeah, here I'd just like to kind of show you the ECTD structure, which we've had in pilot 3. So it features 2 main folders, which is the M1 and the M5. Within this approach, we submitted the REN block file, as well as the pilot 3 utils package. So you can kind of see here, per FDA guidance, when we were, working to generate pilot 3 atoms and such and getting everything packaged up, we asked the FDA where it best to put these files for open source submission, and we put them right in the M5 analysis atom and then the programs folder here, just to share some awareness. And again, this is all publicly available, so you can kind of see where it's at in the case you want to follow the same approach. So yeah, here I want to kind of delve into the findings from pilot 3. So let's start with a computing environment. So we did use a reproducible R environment, which was managed by our REN block file, and the proprietary pilot 3 utils R package, again, which we submitted through the ECTD gateway or portal. Regarding the timeline, the initial submission occurred in August 2023, and then after some back and forth with the FDA, feedback from other team members, we did then resubmit in April of 2024. Excuse me, I think I mistyped that. It should be 2024. And then after the resubmission, we just had some clarifications with the FDA, and after their review, we actually successfully completed our review through the FDA in August of 2024. So some of the issues that we encountered were with relative paths, incorrect package names in our package, which we definitely went back and corrected to full paths and accurate names. So specifically, for instance, we provided the full path to read in SCTM data to generate the atoms, and then the full path to where the atoms were generated, so then we could generate the TLFs within the ECTD structure. And originally, when we submitted the first time, the name of our proprietary package was just pilot 3, but it should have been pilot 3 utils. So we fixed those in the second resubmission. And then in terms of atom data sets and analysis TLFs, there were some discrepancies noted between pilot 1 and pilot 3. including differences in attributes, data types, and handling of missing values. But these discrepancies we did document in the appendix of our ADRG in Pilot 3, which you can read. Again, and when you scan the QR code, our ADRG is available for you to read. Unfortunately, even though we had these discrepancies through our further investigation, it did reveal that there was actually no impact on the TLF for the overall analyses. But it is good to show that, just to ensure that processes that we follow are in compliant with what we've done in the past and are in compliant with CDISC and such. And so attributes like this, just make sure that when submitting an R, things are met to the standards as we know in the pharma industry today. All right, so yeah, so now we're going into pilot 4, and this is a bit of an overview on that. So after the pilot 4 originally was submitted, I think in September of this year, it was received and the review process has commenced. So far, the FDA has noted no significant issues with running the application on the web through WebAssembly, apart from, I think minor issues with a header from the primary tables were switched, but I think that was already fixed. And Eric Nance, probably on the call, is actually the lead for Pilot 4 and has worked tremendously on this to try to get this to work, and has had it successfully work. And so it's really great to see that we're moving into this new WebAssembly and container technology. Pilot 4 definitely builds upon what we've done in the previous pilots. So essentially what it is just with the WebAssembly technology, the FDA are able to then review what was actually done in Pilot 2, the Shiny app, through the WebAssembly regeneration on their end. So that showed to be successful through FDA review. But one thing to note through the agency, it's important to note that Shiny application should be a supplementary tool, not a replacement for existing analysis programs, as emphasized in Pilot 2. So yeah, again, though there is that feedback, the technology and process here was a good to know that we're able to have FDA reproduce on their end. All right, so some of the findings here. So again, after initial review, it was received and reviewed already, and has definitely started. Again, no significant issues. Again, as I mentioned, column headers were switched. So yeah, all of this was summarized in a blog recently, which I have linked here as well. And on top of that, again, is a QR code to the repo for you to review in the public domain here. And then looking forward to actually, with the WebAssembly, the next step for Pilot 4 then is also submit as containers. So yeah, so to conclude our talk, I just wanted to conclude with our achievements and of course our future plans. So we definitely reached a significant milestone by submitting 4 pilots to the FDA via the ECTD portal. And of these, 3 have been FDA approved, successfully completed. And again, Pilot 4 is currently under active review. So throughout our journey, we focused on simplifying the submission processes. We're definitely looking forward to planning larger submissions and exploring dataset integration with JSON, along with many other ambitious goals here. So yeah, thank you for your attention. We look forward to sharing more advances with you in the future. At this time, we just want to say thank you for all of the for tuning in, and if you have any further questions, Q&A and such, feel free to tag us here on GitHub. This is a QR code. You can submit questions through our GitHub issue.
Well, thank you so very much, Ben and Joel, for this amazing summary of what has been years of very intense, but yeah, very valuable effort. So kudos to both of you for being a part of this working group. It has been one of my favorite external adventures I've ever had, and we've learned so much from each other. And I hope this has inspired others to hopefully get involved with our efforts down the road.